Okay, so here's chapter 15 on microbial mechanisms of pathogenicity. So really, it's about how do microbes actually cause disease? What is it that they do to cause disease to us? So you have some terminology here, and I am going to be following along with the study guide. So I think virulence factor, well, although I did not list actual terminology on the study guide, I mean, you're going to see that you need to know examples of virulence factors. So obviously it's going to help to know what a virulence factor is. I think that's the only one that really warrants us defining, because we've been using pathogenicity or pathogen or pathogenic a lot throughout the semester, as well as virulence is another term we've used quite a bit. But a virulence factor is a component, some part of a microbe that allows the microbe to invade our tissues and to take up residence or colonize us, and to be able to adapt to live within our bodies and to be able to overcome our defenses. So it's a variety of different things that microbes possess or that microbes make that can be virulence factors. And some microbes can have multiple virulence factors. Some may only have one, and then some have none. But usually if a microbe does not have a virulence factor, then it's not usually very good or even able at all to cause disease to us, because it really is these virulence factors that contribute to the pathogenicity or virulence, to the ability to cause disease. So before we get into the actual virulence factors, we'll just kind of quickly walk through the things that are required, kind of the prerequisites of getting an infection. So number one, obviously the microbe has to be able to get into your body. And so those entrance points that it uses are called portals of entry. So microbes can come in our mucous membranes, which line our various orifices. The most common mucous membrane that people know about is the one that lines our respiratory tract, but it does kind of have double duty as well, as it lines our upper digestive tract. You have kind of the overlap of the respiratory digestive, sharing the mouth, throat, and all of that. So you've got mucous membranes of the respiratory tract, you've got mucous membranes of the GI or gastrointestinal tract, you've got your conjunctiva of your eyes, which is also considered a mucous membrane, your genitourinary tract, your genitals have mucous membrane. Those are the main ones, I think, that that are in the body and that we will talk about. So mucous membranes definitely can be a portal of entry. Skin can be a portal of entry, but it's not the best portal of entry. Skin is pretty inhibitory to microbes. You've got tightly packed epithelial cells. You've got keratin, which is a protective protein. You've got the high acidity, the high salt. So not a lot of microbes like the skin. And when they do use it as a portal of entry, they usually do have to be somewhat specialized to be able to do that, because we're not talking about a cut or a wound. When we talk about skin as a portal of entry, we are saying they are coming through unbroken skin. These are microbes that can enter into your pores, your sweat glands, your hair follicles, and lead to an infection. And not a lot of microbes can do that, actually, which is good, because again, our skin has a lot of protective things to it. Parenteral route, now this is where there's some break in the skin or other areas of the body, but mostly we're talking about, some kind of wound, burn, cut, needle stick, surgery, anything where a microbe could be deposited directly into your tissues. This is parenteral route. And most pathogens have a preferred portal of entry. And if I were to ask you what's the most common portal of entry, I mean, just even speculating on what we know about disease, I think most, especially what's going on right now, most would probably answer the respiratory tract. Mucous membranes of the respiratory tract is the most common portal of entry. We have to breathe, and when we breathe, we can breathe in pathogens. I don't think you need to know second most common, third most common, so on, but I would say in general, second most common is probably your your gastrointestinal, because again, we have to eat, we have to drink, we have to swallow, and kind of also the fact that they do share the same opening with the respiratory tract. You can obviously see how gastrointestinal would be a pretty common portal of entry as well. So this question, easiest, most frequent traveled portal of entry, respiratory tract. All right, so just because a microbe comes into a portal of entry that it needs to come into, because again, remember, they do have often a preference. They can't just come into any portal of entry. So once they get in the right portal of entry, it does not guarantee that you will get an infection. There's a lot of factors that come into play with that, but one of them is the number of invading microbes, which is a term, and actually kind of a mathematical measurement called the ID50, which the ID stands for infectious dose, and 50 refers to 50% of a sample population. So they've done studies with laboratory animals, and they've determined the ID50 for a variety of diseases. And so the ID50 is a measure of, it says virulence, but I would really more say contagiousness of a microbe. So for example, measles virus, and I'd be curious to see if they come out with an ID50 for coronavirus. I don't know of one right now, but measles virus, they have an ID50 has been calculated to be as low as 1 virus. So what that means is that it can take as little as inhalation of 1 measles virus to lead to measles. And that means it's very contagious. Other microbes may have much higher ID50s. You may have to inhale 50,000 virus, particles before you get infected. I mean, it's quite variable, but you can see how the lower the number, obviously the more infectious or the more contagious that microbe will be. LD50 refers to toxins, which we will cover in this chapter. And so this is the lethal dose for 50% of a test population. So again, on laboratory animals, they have injected various concentrations, amounts of toxin into laboratory animals and determined this LD50 number based on, how much does it take basically to kill off half of the population. So again, the lower the number, the more deadly the toxin is, the more potent or toxic it is. So here's a good example for portal of entry and ID50 kind of combined. So generally a lot of microbes really only have one portal of entry, one preferred portal of entry that they can use. Like, for example, influenza only uses the respiratory mucosa. If you accidentally swallow, which you probably do if you have the flu, you'll end up swallowing some flu virus and it will go into your gastrointestinal tract. You cannot get the flu from the virus going to your GI tract. You don't have the proper attachment factors for the virus there. It's a respiratory virus. It attaches to respiratory epithelial in the mucous membranes of the respiratory tract. But some pathogens do have multiple portals of entry, and Bacillus anthracis is one that does. And it's kind of interesting because not only do you have these different portals of entry that it can take, but you have different ID50s depending on the portal of entry. So notice if anthrax endospores go in through the skin, and again, this is unbroken skin, into your pores, sweat glands, hair follicles, it only takes 10 to 50 endospores. But if you inhale the endospores, you would have to inhale 10 to 20,000 before you get anthrax. And if you ingest the endospores, you would have to ingest 250,000 to 1 million. So you can see the ID50 can vary based on the portal of entry. And the reason for this, I think some of you might know or maybe speculate, but just think about those different areas. What's in our, we'll start at the bottom with the highest ID50. What does that endospore have to survive through to be able to cause us gastrointestinal anthrax? Well, it has to get through our stomach and our stomach acid. And so that's why the number is so high for ingestion, because our stomach acid will destroy a lot of things. So you need to get a really high dose in if you were to actually get gastrointestinal anthrax. Well, what about inhalation? Well, we have some protective mechanisms in our respiratory tract, our ciliary escalator, our mucus. And then actually once, if the microbe makes it to our lungs, in our lungs, we have white blood cells that are fixed there. They're called macrophages, fixed macrophages. So we have protective mechanisms. So it, takes a higher amount, but not as much as GI. And then look at the low portal of entry for skin. Well, it's because we don't have a lot of circulating white blood cells in our skin. We have very few, only a few specialized types of white blood cells can you really even find in the skin. So it doesn't take much really to get cutaneous anthrax. Good news is cutaneous anthrax is the most mild. Pulmonary inhalation anthrax is very fatal, and GI is somewhere in between. So what about LD50? Well, kind of like ID50, the lower the number, the worse it is. The more for ID50, the more contagious or infectious it is. For LD50, the more lethal or the more potent this toxin is. So you could compare botulinum toxin involved in botulism, Shiga toxin, and staph enterotoxin, which is involved in staph food poisoning. And you can see then, looking at the numbers, that botulinum toxin is the most toxic. It only takes a tiny, tiny quantity to lead to botulism, which can be fatal because it's a neurotoxin. It takes a lot more of the food poisoning toxin, staph enterotoxin, to lead to food poisoning. So here's a question. The LD50 of botulinum toxin is 0.03 nanograms per kilogram. And then the LD50 of salmonella is 12 milligrams per kilogram. So that's why you have the conversion there. That threw me off for a minute. But you can see with all those zeros, right, that there's quite a big difference if we just use the milligram per kilogram. The botulinum toxin takes just a tiny quantity versus salmonella, which would take a lot more to be lethal. So our answer would be the botulinum toxin is more potent. Okay, so once a microbe gets in, has enough ID50 to stick around, well then what happens? Well then it adheres. It has to attach to something, right? Usually some receptor on your cells. And so microbes do have some special things that help them attach, and these also can be considered then virulence factors because they're involved in that microbe being able to be virulent, to be able to cause disease to us. So you've got like the glycocalyx, which we talked about before, which either could be in the form of a capsule or a slime layer. Fimbriae, biofilm formation in itself is definitely a virulence factor and is involved in attachment of microbes. Here's just some diagrams, just kind of showing, okay, usually there's some kind of binding of some receptor on the host cell and some kind of adhesin or ligand, some component of the microbe on its surface. If it's a virus, it might be a spike, right? If it's bacteria, it might be a capsule or a cell wall component if they don't have a capsule. So this is showing E. coli. It uses fimbriae to attach to our intestinal cells, but like we were actually talking about today in our chapter 14 lecture, if E. coli gets into our bladder, now it can cause a bladder infection, and it's going to use those same fimbriae to attach to the bladder cells. Here's it just says bacteria purple, but definitely looks like a Staphylococcus to me, which are very common on our skin. So just quickly through these slides on virulence factors, it is number 3 on the study guide, examples of virulence factors. So definitely just being able to recognize what are the different possible virulence factors, and maybe just a little characteristic of each. One that we've already covered since the beginning pretty much of the semester was capsules, which we know can help the bacteria attach, but also they can prevent phagocytosis. So bacteria that have capsules tend to be more virulent or more pathogenic. But it's not just capsules, because some bacteria don't have capsules, but they still have the ability to attach to our cells and to avoid destruction by our immune cells. So you have some other cell wall components like M protein, the OPA protein, mycolic acid, which is found in mycobacteria. And then once the bacteria do attach to our cells and kind of invade our tissues, or are starting the invasion process, they can be making various substances, especially enzymes, that can help them in that invasion process or in that infection process. So these are also virulence factors. They're just listed on number 3 of the study guide as enzymes, coagulases, kinases, hyaluronidase, collagenase, and DNase. And so you do want to know this slide, know these enzymes, and be able to recognize what each one does. In general, you'll see they're definitely affecting our tissues, especially like the hyaluronidase, the collagenase, the kinase. These ones are breaking down connective tissue, digesting blood clots where our body may have wanted to try to contain the infection, and the bacteria is able to spread because of production of these enzymes. DNase as well, because once the bacteria maybe kills our cells, then our DNA spills out and gets in the way. So bacteria are able to break down even our DNA as well. And then coagulase is the only one that is a little bit opposite of these that are involved in breaking down. Coagulases actually form a clot. They coagulate blood. Bacteria that make coagulases, the main example is Staphylococcus aureus. And it actually makes sense when you look at the type of infections that Staph aureus causes. It mostly causes boils, abscesses, which are a localized infection, a walled-off infection. And the characteristic of that infection really is contributed to the production of coagulases. The bacteria kind of walls itself off to keep immune cells out, and that's why it really forms that little kind of concentrated, localized infection versus something like Strep pyogenes, which causes flesh-eating disease. It makes these enzymes, the kinases. It actually has special ones called streptokinases. It makes hyaluronidase and collagenase. And so that's why with the strep, especially that flesh-eating or necrotizing fasciitis infection, why you see destruction of tissues is due to the production of these enzymes. So to keep the video pretty short, I'm going to skip the animations. One last thing I want to point out about virulence factors before I'm going to be stopping this video, and I'll make a separate one on toxins. But one last thing is antigenic variation. So it's also on the study guide under number 3. So just be aware that this is a virulence factor. The fact that microbes can mutate and they can change their antigens, which we will be covering in future chapters 16, 17. Antigens are the kind of the foreign material, some kind of substance on the surface of a microbe that our immune system recognizes with antibodies. So we have this way of our immune system to recognize a pathogen. Well, what does the pathogen then do in response to that? It changes its surface, right? So that then it changes the ability of our immune system to recognize the pathogen. So some pathogens can mutate very rapidly on a regular basis, and it makes it really hard for our immune system to be able to then destroy that pathogen because they can't keep up if it keeps mutating. So I'm going to skip this other stuff on the invasins. You don't really need to know about those. We already mentioned biofilms definitely play roles in virulence. What's the action of bacterial kinases? So this one should be to break down fibrin, right? Siderophores are another virulence factor that I don't think, it doesn't look like it's on the study guide, but just brief mention if you're interested. I put the chemical structure here, and I put this interesting link here. Definitely has a lot of chemistry in it. You can see bioinorganic chemistry readings, but it still is, I found it really interesting, so hopefully the link still works there. But it's basically a substance that bacteria make to steal iron from us. And so these definitely are virulence factors, and they can play major roles in disease symptoms of certain infections. When we are robbed of iron, it really can affect your, functions in your body and in your immune functions, especially. So we don't really need to know any details here. It's just kind of, mentioning different direct damage that can occur from the presence of microbes in our bodies. But the next topic, which I will make a separate video on, is toxins. And that's all going to be encompass number 4 on the Chapter 15 study guide.